Okay, so good evening, everyone. So today we are in the week 12 problem solving session of this NPTEL course Molecular Biology. So today is the last problem solving session for this course, and I hope all of you have gone through the week 12 lectures. So the week 12 lectures deals mainly with the applications of molecular biology. So starting from how different enzymes are used in different industries, starting from dairy industry to bread industry, bakeries, then so what are the different enzymes that are required? Then then the topic that was discussed was genome editing, how genome editing is being used for creating transgenic animals, and some PCR based techniques, how are they being used and what are their applications. So we will go through these topics one by one. Also, I will solve a few sample questions that will help you to understand the concepts better. So I'll wait for a few minutes, 4 to 5 minutes, and then I'll start today's session. So let other learners join. Okay, so I will start today's session. Let me share today's slides. Okay, so yeah, good evening, everyone. So I welcome you all to the week 12 problem solving session for this NPTEL course, Molecular Biology. This course is being run by Professor Vishal Trivedi of IIT Guwahati, and I am one of the PMRF TAs for this course. I'm Akash Jyoti Pathak, and I'm a PhD scholar in the Department of Biosciences and Bioengineering. So let's have a quick recap of what all we have covered in week 12. So in week 12, the topic that were covered it mainly dealt with applications of molecular biology. So we looked into or we understood what are the how molecular biology is being used in genetic engineering, in PCR-based application, in creating transgenic animals, and in genome editing. So in genome editing, you already we discussed the genome editing in detail in the extra session as well as in week 11 as well. So in genome editing, there are three main techniques that were discussed. One was the zinc finger nucleases. How zinc finger nucleases are used, then talents, transcription, activator like effector, nucleases, talents, and then the most the most widely used or recently the most widely used technique that is being used is CRISPR-Cas. Okay, so let's discuss and understand some of these concepts through this lecture. So the first question for today is the enzyme utilized to enhance flavor development by breaking down proteins into smaller peptides and amino acids. So try to answer this question. So the options are protease, amylase, lipase, and lactase. So this is quite an easy question because it is you do not require need you do not need much knowledge for answering this question because obviously you know that for breaking down DNA there are DNases, for breaking down RNA there are RNases, right? And for breaking down carbohydrate, what are used? Amylases. For breaking down fats or lipids, the enzyme that is used are lipases, isn't it? Okay, so and for proteins, what are the enzymes? So they are proteases. Okay, so this is basic biology and you all know about this. Okay, so but just by reading the second part of the question, that breaking down proteins into smaller peptides and amino acids, you can answer this question. So the answer is protease. Okay, option A. So let's understand how these proteases are used to enhance flavor. Uh, in and it is used in the food industry, how it is being used. Okay, so proteolysis, so breaking down proteins into smaller peptides and amino acids. This process is called proteolysis, isn't it? So this proteolysis is crucial in various food industries, including dairy, meat, bakery, to improve texture, tenderness, and enhance flavor. Okay, so in the meat industry, it is mainly used for making the meat tender and soft, because these proteases will act on protein and break it down into smaller polypeptides, which will be further broken down into amino acids, individual amino acids. Okay, so yeah, this is directly from the lecture slide. So this protease is widely distributed in biological. This protease is widely distributed in biological world. You can find it. So these proteases are also present in the cytoplasm. Okay, so if you lyse the cell, these enzymes are very prevalent. There are different types of proteases, serine proteases, then other kinds of proteases are present. So they're widely distributed in biological world, and it hydrolyzes the specific peptide bond to generate para-K casein and macropeptides in production of cheese. So not only in production of cheese, it is also used for production of meat, in production of uh, different fruit juices. Okay, so it is widely used. This enzyme is widely used, and they are also used in cheese and yogurt production to alter protein structure, influencing the final texture and flavor of the products. Okay. So not only it also so what it has two advantages. These proteases, it improves the texture, and also what it does, it adds flavor to the products. Okay. So how do they add flavor? Let's understand now. So there are this figure is taken from this paper. So this paper explains how different type of proteases are used. So you can see in the figure on the right, there are different types of proteases. There are endopeptidases, then there are different dipeptidyl peptidases, tripeptidyl peptidases. So there are different type of tripeptidases. So tripeptidases, it will break down tripeptide into dipeptide and a monopeptide. Then dipeptidases will break down dipeptides into two amino acid residues. Okay, so there are carboxypeptidase, amino peptidase. Now these peptidases, they are very specific as they act at specific locations or in the amino acid chain. Okay, so there are specific amino acid residues which are recognized by the carboxypeptidases and amino peptidases, and some of these peptidases act at the N terminal of a particular amino acid, some act at the C terminal of a particular amino acids. So you must have learned about this in biochemistry as well. So yeah, so the research emphasized that bitterness is a major limitation in food industry, which limits the consumption and utilization of food protein hydrolysates. Now, aminopeptidases are capable of reducing the bitterness. So among the proteases, which protease is mainly responsible for reducing the bitterness, they are aminopeptidases. Okay. So how do they reduce the bitterness? So they increase the degree of hydrolysis, thus releasing free amino acid. Okay. So the degree of hydrolysis and free fatty acid content were increased when treated with recombinant aminopeptidase from this particular species. Okay, thus it reduced the bitternesses of casein and soy protein hydrolysates. So, this is the food protein hydrolysate, and when this is acted on by endopeptidases, it is broken down into polypeptides and peptides. Okay, then these polypeptides and peptides are acted on by these other peptidases, dipeptidyl peptidases, tripeptidyl peptidases. Then this X-proline dipeptidases, which breaks it down into bioactive peptides. So these are small, even further smaller peptides. These are smaller peptides, and then these are acted by these type of peptidases, mainly the aminopeptidases, which breaks it down into smaller, which breaks down the smaller peptides into free amino acid residues. Okay, now beta glucanase improves beer quality by the options are breaking down proteins, hydrolyzing fats, reducing beta glucan content, and enhancing aroma. Okay, so these are all fact-based questions. You need to know what are the different enzymes which are currently being used in food industry. Okay, so try to answer this question. So as we can also directly answer by reading the options as well. So beta glucanase will obviously act on beta glucan. Okay. Mhm. So it will act on beta glucan, and it will reduce the beta glucan content that is present in beer. So this let's understand what is this beta glucan. Okay, so you can see here this was explained in the lecture as well. So the beta glucanase represents a group of carbohydrate enzymes. Okay, so what is another carbohydrate enzyme? A common carbohydrate enzyme is amylase. So similar to this carbohydrate enzyme, so amylase act on amylose, right? So beta glucanase will act on beta glucan, okay, and break down glycosidic bonds within the beta glucan. So, and it also aids in filtration after mashing and brewing. So, this barley that is used, uh, which is the substrate, or which is the raw product from raw material from which the beer or alcohol is obtained. So this undergoes several processing. You can see here in this figure, it has been shown what are the different processes that take place in a brewing industry. So these are milling, mashing, lautering, boiling, whirlpooling. So these are, then there is a fermentation tank and filtering. So this in this filtering process also, this beta glucanase is mainly used. Okay. Beta glucanase enzyme. So there are a series of steps which is required for getting the final product. Okay. So the beta glucans, it is a type of polysaccharide. It is a type of polysaccharide which is found in barley, which can negatively impact beer filtration and overall quality. Okay. So the beta glucan is so basically as amylose is also is a polysaccharide, you know. So the similarly the beta glucan is a polysaccharide. Beta glucan is a polysaccharide and it is mainly present in barley. And this negatively impacts the beer filtration and overall quality. So this once this beta glucanase is added, it improves the beer quality by breaking down the beta glucans. So these glycosidic bonds which are present between the monosaccharide subunits, these are broken down, and thus what will it do? It will help in reducing the viscosity of the wort. So the liquid that is extracted from the malt. So the malt is the barley that is processed and that is used, and it enhances filtration, improves wort yield, and leads to clearer, more consistent beer. The beta glucans are a major component of barley cell walls and contribute to the viscosity of the wort, making it difficult to filter. So these beta glucans are present in the cell wall. They are present in the cell wall of barley. So this beta glucanase breaks down this beta glucan, and it does helps in reducing the viscosity and improving the flow of water during filtration. Okay. I hope this is clear. Now, this third question is asking that what is the main hallmark of Alzheimer's disease pathology observed post-mortem? Okay, what are the options? They are tau protein tangles, amyloid plaques, degraded hemoglobin, and neurofibrillar tangles. So, many of you might have read about Alzheimer's disease, and this is a very widespread or uh, this is in many research labs are working to solve this problem of Alzheimer's disease, which are working on this Alzheimer's disease. So uh, so yeah, this is the what is the answer for this question? So it is amyloid plaques. Okay. So Alzheimer's disease, as you know that what happens in Alzheimer's disease at with age, uh, people suffer from this disease, and this disease is associated with forgetfulness. So people forget the name of their or name of their the persons they know, or they can forget what are the common daily work they have to do. So such type of problems are associated with this disease. And so this concept was discussed in the lecture as well. So it is a neurodegenerative disease, isn't it? So neurodegenerative means the neurons which are present in the brain, they with age, these neurons get they get degenerated. And also it is mainly associated with the formation of amyloid plaques. So amyloid plaques, they are composed of this amyloid beta peptide, A beta peptide, and A beta peptide fragment of beta amyloid precursor protein. Okay. So for Alzheimer's, as you can see, it has been described in this slide as well. So this Alzheimer's disease is a progressive disease which causes progressive brain damage, causing problems with memory, thinking, and behavior. So yes, that is what I was explaining. That so it's associated with memory, and how does this disease progress? So this disease, you can see that the ventricles which are present in the brain, in those ventricles, there is accumulation of these uh, of plaque and tangles. So these are commonly called as, so they are basically filamentous protein which aggregate and form fibrillar structures. So this leads to the loss of connections between the nerve cells and eventually cause the death of nerve cells and loss of brain tissue. So if the nerve cells are lost, then then what will happen? Then you will then the person will face difficulty in doing common task or in recognizing someone or remembering something. So there will be difficulty. So the patients will have a shortage of chemical messengers, which is important for transmitting signals around the brain. So because these nerve fibers are damaged, and also the connection which is present between the different neurons, they also get damaged or get degenerated, and the plaque starts building up in the ventricles of the brain. So all these problems results in this disease, Alzheimer's disease. So APP mutation, so this is a beta amyloid precursor protein, and the mutation in this protein induces peptide aggregation. So for doing research in Alzheimer's disease or finding a cure for this disease, there are transgenic mice models which are called Alzheimer's mice. So this transgenic mice is named as TG2576, and it is a why it is a this transgenic mice. How has it been developed? It has been developed to produce genuine human amyloid. And this is the world's first Alzheimer's mouse incorporated an EPP mutation. So this protein has been mutated, and the mutation of this protein results in the formation of this amyloid plaques. The pathophysiology effects are similar in transgenic mice and comparable to humans. So if you want to test any drug against this disease, so you cannot directly apply them on humans. So you have to first test them on mice. So for this reason, there are different mice models. So one of them is this one. So for different diseases, there are specific mice models which have certain mutations or certain uh, deletions in the gene which make them susceptible to a particular disease, and thus can mimic the disease which are commonly affect human beings. Okay. So on applying the drug, you can check whether these for example, have developed a drug which can act against which can be a possible cure for Alzheimer's. Then then you will see that on treating the mice, you have to take the brain cells and see how this plaque buildup, whether they are decreasing or how is the morphology of these plaques changing. So such type of research are going all over the world. So yeah, let's understand this disease. So Alzheimer's disease. Uh, so as this disease progresses, just give me a second. Okay, so you can see this figure. So this figure has been taken from this paper, and this paper describes that how this disease progresses. So I was talking about this protein, APP protein, which gets mutated and leads to the plaque buildup. So what happens is as the disease progresses, the brain tissue shrinks. So this is a healthy brain. You can see in this figure that this is the image of a healthy brain, and as the disease progresses, the size of this brain shrinks. So this is the Alzheimer disease brain, and so what happens? The volume of the brain also decreases. Volume of these ventricles. So ventricles are basically the spaces which are present in the brain and which remains filled with the cerebrospinal fluid. So this volume, the volume of the ventricle increases. So you can see here that the ventricles in a normal healthy brain is quite reduced, while this volume of ventricle in an Alzheimer disease patient is enlarged. Okay. And then there are amyloid beta peptides. So the amyloid beta peptides are produced by the cleavage of the amyloid precursor protein. So APP is what? It is a amyloid precursor protein, and it is present in the membrane of neurons. So this is the structure of a neuron. You all must have seen the structure of a neuron. It has axon and dendrites, uh, the dendrites and axon, and the membrane of the neuron. These proteins are present. Amyloid precursor protein. Okay, and the so this protein, this precursor protein is cleaved. This precursor protein is cleaved, and what happens after that? From this precursor, then amyloid beta particles are amyloid beta peptides. Amyloid, so there is amyloid peptide precursor protein, amyloid precursor protein. So they are cleaved and uncleaved. Once this amyloid precursor protein is cleaved, it forms amyloid beta peptides. It forms amyloid beta peptides. Now these amyloid beta peptides undergoes aggregation. Okay. So in the space between the neurons, the amyloid beta forms oligomers. Okay, they form oligomers. So one amyloid beta peptide joins with another, and they associate with each other to form an oligomer. And that is thought to disrupt the function of the synapses and receptors present in the neuronal plasma membrane. So you see here that on forming oligomer, they are starting to build up as the disease progresses, and they get accumulated in the neuronal tissue, and thus prevents the chemical messenger from being transmitted from one neuron to another, and also it leads to the death of these neuronal cells. So the fibrils of the amyloid beta oligomers in plaques interfere with the function of neurons. So what is the function of neurons? They are involved in transmission of information or nerve impulses from one part of the brain to the other. So when these plaques are formed, this function will get impaired. And also, finally, what happens? There is tau hyperphosphorylation, which causes neurofibrillary tangles within neurons, displacing intracellular organelles and disrupting vesicular transport. So these neurofibrillar tangles are formed these tau proteins. These tau proteins again get associated with each other, and these neurofibrillar tangles starts to build up in the cytosol of the neuron. And this disrupts the vesicular transport. So the vesicles contain the chemical messengers. So these chemical messengers are now not able to move from the cell body or from the dendrites to the axons, from the cell body to the axons. Okay. Now let's understand what are the transgenic animals. So how are transgenic animals created? So what is the meaning of transgenic? The transgenic means in the cells of these animals, it will have a particular mutation. So in all the cells of this animal, there will be a mutation. So if you want to study what is the function of a gene, or uh, what will happen if the gene is deleted, how the deletion of a gene affects the organism, then what you can do, you can cause a knockout of the gene and then study how this animal is behaving. So these are transgenic animals. So the mice models that are produced, they are also that are produced for studying the different diseases. They are also produced in this way. So So this the basis is microinjection of DNA. So these transgenic animals they contain a cloned gene in all of its cells, and they are generated by microinjection of a fertilized egg cell. So this the concept has been taken from the book T.A. Brown. Second go, this is a very standard classical textbook which is followed. And so this is generated by microinjection of a fertilized egg cell, which is subsequently cultured in vitro for several cell divisions and then implanted into a foster mother. Okay, so we will understand this how this microinjection takes place in the next slide. So the knockout mice used to study the function of human and other mammalian genes are examples of transgenic animals. So if you have so knockout mice, you must have read about these knockout mice in different research papers. So they are basically used to study the function of humans and other mammalian genes. So how will you know if a particular gene is involved in a specific phenotype or a specific function? You have to, there are two ways. Either you delete that gene and see what is the effect, or you can mutate it and see what are the changes or that is getting that is, or if you know that yes, a gene or when mutated produces a particular phenotype, you can check how this how this takes place by creating this knockout mice. So although this technique works well with mice, the injection of the fertilized cell is inefficient or impossible with many other mammals, and the generation of transgenic mice or animals for recombinant protein production usually involves a more sophisticated procedure called nuclear transfer. Okay, so this method is actually very it requires a good expertise or a good hand. So this is uh, it does not ensure that this will generate transgenic mice, and this is a costly method and not an easy method. So for this, what another technique is used, which is called nuclear transfer. So how does this nuclear transfer takes place? So this involves microinjection of the recombinant protein gene into a somatic cell. Okay. So because the somatic cell will not itself differentiate into an animal, its nucleus after microinjection must be transferred to an oocyte whose own nucleus has been removed. Okay. So now, now the difficulty of using or micro-injecting a DNA into an oocyte. So if you inject a DNA or a nucleus in an oocyte, then what will happen? Then that nucleus, the nucleus that you have injected into an oocyte, those two nuclei will fuse and it will give rise to zygote. And then there will be several processes taking place, uh, which will not uh, which or which will not ensure whether whether the transgene that we have in the nucleus, whether it will be present in the zygote or the embryo being formed. Okay. So for this process, what is done? The somatic, this DNA is injected into a somatic cell, in the nucleus of a somatic cell, and then this somatic cell is allowed to grow. Okay, so once this somatic cell will grow, then you take out the nucleus from this somatic cell and inject it into the oocyte. So the oocyte, what you have to do, you have to remove the nucleus from the original nucleus from the oocyte and inject the other nucleus. Okay, so because the somatic cell will not itself differentiate into an animal, so oocyte will have has the property of totipotency, and it will differentiate into an animal. Okay, it will start dividing from zygote, embryo, and it will subsequently divide. But the somatic cell will not differentiate. It does not have the ability to differentiate. It will only divide. Okay, so this nucleus is taken out, and then and then it is transferred to an oocyte whose nucleus has been removed. So after the implantation into a now this oocyte, then oocyte, this oocyte is then implanted into a foster mother. Okay, the engineered cells retain the ability of the original oocyte to divide and differentiate into an animal, one that will contain the transgene in every cell. Okay. So once a transgenic animal has been made, it can reproduce and pass its clone gene to its offspring according to the standard Mendelian principles. So now there is a question. So this transgenic animals was discussed in detail in the lectures. So answer this question. The transgenic animal that has been engineered for potential use in organ transplantation to human is transgenic chicken, transgenic pig, transgenic rabbit, transgenic mice. So you might have read about various news uh, that about these things also about the organ transplantation, how so xenograft, how organ from one species is engineered or is engineered so that it can be transferred and not rejected by another organism of another species. So this is again a fact-based question. This is the answer is transgenic pigs. Okay, so these transgenic animals, they are genetically modified to have organs that can be transplanted into humans. And how they have been engineered? So they have been engineered in a way that they do not express key foreign antigens. So you know about the different MHC class antigen. So there is MHC mismatch. So which results in rejection of the of any foreign substance that is injected into a host organism. So these foreign antigens that are present on the organ, uh, these are these are knocked out, or these genes are knocked out so that such type of proteins are not expressed, and hence they will not cause an or elicit an immune response. So these are promising alternative to human donors, right? So pig is the only animal used. So remember this point that pig is the only animal used for organ transplantation. And the porcine heart valves successfully transplanted into human heart. There are liver, lungs, kidneys. They are also being tested, but the one that has been successful is the heart that has been transferred from a pig to a human, and that has been successfully taken up or not rejected by the host human. Okay. So in physiology, why is it so? Because its physiology closely matches that of humans and it is much less expensive than monkeys and other primates. Okay. So monkeys are more closer to humans. The ancestors, they are ancestors of humans, but creating by transgenic monkeys are costly, and so these transgenic pigs are used instead. Mhm. And so this is what I was discussing. So the specific modification that is typically introduced into transgenic pigs to reduce the risk of organ rejection in human recipients is so knockout of major histocompatibility complex, introduction of human leukocyte antigens, then overexpression of xenotransplantation antigens and knockout of the complement system. So what will be the correct answer for this question? So you all must have read about this major histocompatibility complex. So what are these? So they are a group of genes that code for proteins that help the immune system identify self and non-self. So these are genes basically. So they are a group of genes and they will code for a protein that will help that will help in distinction between self and non-self. Okay. So because the organs which is present or the tissues which are present, they all have these different types of MHC class proteins and these the MHC class proteins which are present in the tissues, they are self MHC class molecules, but that are present in other organisms, they will have different type of proteins. So that will cause an immune reaction. Okay. So how this is done? So the answer is yes, option number B actually. Okay. So why option B? Because what has been seen in recent xenotransplantation research, especially with transgenic pigs being prepared for organ transplantation into humans, scientists are not only knocking out pig genes that trigger immune reactions, but also introducing specific human genes, including human leukocyte antigen and immune modulatory proteins that actively prevent immune rejection. Okay, so both the things have to be done actually. The major histocompatibility complex proteins that are present in the that are specific to the pig or specific to a particular organisms, they have to be knocked out, and the human leukocyte antigens. So these are the MHC molecules which are particularly present in humans. Okay, so these also has to be introduced so that the chances of rejection, organ rejection decreases. So this point is clear. So these are basically this. So this MHC class of antigens, these are specifically they are a particular gene loci. Group of genes. They are group of genes which are present in a particular gene loci. Okay, and what is their function? They express a type of glycoprotein. Glycoprotein molecules. Glycoprotein molecules, and these glycoprotein molecules, they are responsible for rejection of grafts between genetically unidentical individuals. Okay. And in humans, this major histocompatibility complex, these are known as human leukocyte antigen. So in humans, this MHC are known as human leukocyte antigen or HLA. And there are three different, so you might have been reading about this in immunology. There are three common MHC molecules. These are class 1, class 2, and class 3. And the genes of this MHC exhibit genetic variability. And the MHC has several genes for each class. Hence, this MHC is also very polymorphic or polygenic. Uh, it is highly polygenic, means the sequence varies in different individuals. That is why you cannot transfer organ from any individual, not only between not only between species, but also if you transfer an organ from one human to another human, that also gets rejected because of this MHC molecules, because they are very polygenic and they are different individuals. Humans have different type of these glycoproteins that are expressed. Okay. You will read about all these in immunology as well. Okay. Now answer this question that genetically modified smart mice are often used to study. So why are they called smart mice? So if you want to study about any neurological disorders or whether or any brain function of mice, then there has to be some transgenic mice, genetically modified transgenic mice. So these are basically called smart mice. There are different types of there are diverse research areas that deals with how what is the function of a particular brain, or how the brain is involved in remembering things, or memory, or how memory builds up, on these or cognitive behavior, how the behavior of the brain can be are currently in research. So for these type of things, you need there are these genetically modified smart mice. So these genetically modified smart mice, they have this advantage of, or they are modified in a way that they can learn and they have memory. Okay, so they have superior learning and memory. So one of this mice which has this ability are the Doogie mice, and it is modified to overexpress this particular transgene NR2B in the cortex and hippocampus, thalamus, brainstem, and cerebellum. So overexpression of this transgene NR2B helps this mice to have superior learning and memory. So these are very interesting topics, and you can read about them in on internet. Yeah, and you can gain interest in these areas. Okay. So answer this question, the molecular technique that is commonly used to discriminate between mutant and wild type alleles in tumor specific mutation is, so you all have read about these different types of PCR techniques. These different PCR techniques were also discussed in one of the earlier problem solving session. This Tagman probe based techniques, cyber green. So in real time PCR, we had talked about the Tagman probe. Okay. And also in real time reverse transcriptase PCR, RT-PCR, these topics were covered. So this how this Tagman probe has two different probes at its two ends. One is a One is a quencher and another is a fluorophore. So when this quencher and fluorophore come together, then you won't observe any signal. But when this probe goes and bind to a specific region of the DNA, then this probe, this is actually hairpin. This has a hairpin shape. So when this opens up, this quencher gets moves away from the fluorophore, and hence when you excite it with a particular wavelength of light, emit a it will emit a particular light which will be in the visible range, and you can detect its signal. Then there are cyber green dye. Cyber green dye, what does it do? So the cyber green dye, they can intercalate between DNA base pairs. So as the DNA molecule will be amplified, and the more number of DNA molecules, and the more signal or the cyber glyphs. The molecular based beacon based techniques, and there are allele specific PCR, which we will learn, which we will understand to this question. So the answer is actually allele specific PCR. So what are these tumor specific mutation? So the tumor specific mutation, a particular gene may get overexpressed, or it may get deleted, or there can be different there are as for example, there are different types of genes that are those are tumor suppressor gene. Then there are oncogene. So if there is deletion of tumor suppressor gene, then that can be a cause for tumor. Or if there is an oncogene, and this pro-oncogene can get converted into oncogene due to some insertion or deletion of particular basis. Okay, so that is what it is talking about. So whether how will you distinguish that 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 whether there has been a mutation in a particular gene which is responsible for this for the formation of the tumor. Okay. So these are allele specific PCRs. So you know what are alleles, right? So they are alternative forms of a gene. They are alternative forms of a gene, which means that these genes are present in homologous chromosome. They're present in homologous chromosome means these genes will be present. So we all have two copies of chromosome, and each gene are present in two copies. One is present in the paternal chromosome, and other one is present on the maternal chromosome. So these genes may be identical, okay, or there may be some variations in the gene. Okay, there may be some variations. So these minor variation or this alternative form of a particular gene is called a allele. So here also, why is it called allele specific? Because this is similar to what we see in tumor specific mutation. So a portion of the gene can get deleted, or it can get inserted, or there can be a single base that gets added. Um, okay, or it can get deleted. So this minor variation which are seen in alleles, they are seen in tumor specific genes as well. So that is why they are called allele specific PCR. Okay, so let's understand this technique. So it is a powerful and widely used molecular technique which is specifically designed. Okay, so it is specifically designed to differentiate between the wild type and mutant alleles of a gene. So mutant alleles of a gene, so in Mendelian genetics also you have learned about this. There's different this capital T and small t. So this capital T is the wild type or the wild type gene which is capable of expressing the wild type protein, while the small t means this gene is mutated. It is not able to express a particular protein, okay, which is resulting in which is rather uh, which is why these are called these minor variations in this portion of the gene results in these being called as alleles. And so this wild type and mutant alleles of the gene can be is valuable in cancer genomics, where tumor-specific mutations are often subtle, such as single nucleotide polymorphisms or point mutation. So specifically, if you want to study the single nucleotide polymorphisms, that is So yes, this allele-specific PCR is valuable in cancer genomics, where tumor-specific mutations are they're, often subtle, and which these tumor-specific mutations which are caused due to a single nucleotide polymorphism or point mutation. Then such type of mutations can be studied by this allele-specific PCR, and it relies on the principle that primers designed to specifically match a particular allele will only amplify that allele, effectively eliminating amplification of other non-matching alleles. So this allows for direct detection and identification of specific gene variants. So when for doing a PCR, what you require a forward primer and a reverse primer. Now this forward primer is specific to one of the strands of the gene, one of the strands of the DNA. It can be specific to the 5 prime. So the primer which is specific to the 5 prime to 3 prime DNA strand, that is called the forward primer, and the primer which is specific or complementary to the 3 prime to 5 prime DNA strand, that is called the reverse primer. So you can design the reverse primer and forward primer in a manner that one of these primers will be specific to the wild type gene. Okay, so the primer which will be specific to the wild type gene, it will go and bind to that portion of the DNA, and that portion will get amplified. While if a primer is this primer that you have designed for this wild type gene, this will fail to bind to the gene which is carrying the mutation. Okay, it is carrying the mutation, so that will not get amplified. So you have for example, you have isolated the DNA, and you have uh, by restriction digestion, you have that particular portion of the gene which you suspect that could be involved in in cancer phenotype or tumor phenotype. So you take a DNA from the normal cell, you take the DNA from the cancer cell, and then you amplify that portion of the gene using the forward primer and reverse primer. So so if this tumor is having that particular mutation, then this forward primer will not be able to bind, and that will not get amplified by this wild type cell or the normal cell, which is not having the mutation, it will go and it will amplify that portion of the gene. Okay. So the code of this allele specific PCR is the design of primer. So the main crux of this technique is how the primers are designed. So if the primers are highly specific to a particular allele or genetic variant, or whether so these primers will have a sequence that perfectly matches a specific DNA sequence. Okay. So including the SNP or other variant of interest, the PCR process amplifies the targeted gene region using the specific primer. So if the primer matches the target allele, it will be amplified successfully. If it doesn't, the amplification will be blocked. Okay. So the amplified products can then be analyzed. So if it is amplified, then you will get when you run an agarose gel electrophoresis, you will observe a band. While if it is not amplified, you will not get any band. Okay, so that is the basis. So you see here, this figure has been taken from this website. This is a Thermo Fisher website. This is which explains how this any specific PCR works. So you have this is the wild type gene. Okay, wild type gene is having so this portion of this gene is having this T base pairing, while the mutant gene is having instead of this AT, it has mutated to this adenine is mutated to guanine. Okay. Now what will happen? Now you have created this primer, which is complementary to this particular region of the gene. So this primer is having A is having so if it is so if your template strand is having T, then your primer will have complementary sequence which is A. Now this primer will is capable of binding to this portion of the gene and amplifying it, but this primer which is having but the same primer will be unable to bind the gene which is having this mutation, this C. Okay, so this will not get amplified. Okay, while this particular gene, there will be amplification exponentially. So now let's talk about zinc finger nucleases. So I've already discussed about zinc finger nucleases in week 11. So let's understand this topic again. So the zinc finger nucleases, they are all these different type of nucleases that you have read. They are all different endonucleases. Okay, so they're all the function of these different tools of this gene editing proteins. They are mostly they are all endonucleases. Or their main function is to they have two main functions. First, they will recognize a portion of the DNA, and after recognizing, they will bind to that portion of the DNA, and then it will cleave it. Okay, so on once it is cleaved, then if a particular gene is mutated, then that gene can be removed by the action of these different nucleases, zinc finger nucleases, or talens, or Cas9 protein that we have heard. They are all different endonucleases. Okay, so it is an artificial endonuclease that consists of a designed zinc finger protein fused to the cleavage domain of the FOC1 restriction enzyme. Okay. Okay, so the main nuclease activity of this protein lies in this domain, which is the FOC1 restriction enzyme. Okay, and the recognition, so the ability of this nuclease to recognize a particular segment of DNA lies in this zinc finger protein. Okay, so the zinc finger protein, so it has two domains basically. One is this zinc finger protein. It recognizes a segment of the DNA, and the fork one will bind and it will cleave that portion of the DNA. Okay, so you see this figure is taken from this Nature Reviews Genetics paper. So you can see here that one of this is having this. So this is a zinc finger nucleus. So there are tandem arrays, or the zinc finger protein are arranged in tandem repeats, and which is binding to that portion of the DNA, and then it is fused to the FOC1 restriction enzyme. So this FOC1 restriction enzyme will bind and cleave that portion of the DNA. So it is designed to cleave new targets by developing zinc finger proteins with new sequence specificities. Okay. Now, for genome engineering, a zinc finger nucleus is targeted to cleave a chosen genomic sequence. What is the your goal or aim? It is to cleave a chosen genomic sequence, and the cleavage event induced by the zinc finger nucleus provokes cellular repair processes that in turn mediate efficient modification of the target locus. So once you get a double-stranded break, then the repair mechanism of the cell will start to function. So repair pathways, there are two repair pathways. One is the non-homologous repair pathway and the homologous repair pathway. So what you can do is, so this was explained in the lectures as well. So if you design a construct which is having homology to the target DNA, then so so this portion of the DNA. So this is the portion of gene. So this is a portion of gene which you want to insert in the cleaved portion of your target DNA. So what you have to prepare, so this left arm and the right arm of this gene must have homology to the target gene, or to the region where this zinc finger nucleus has bound. So once this is homologous to this left and right portion of this DNA, then by a homologous recombination, this portion of gene can get inserted into the your target gene. Okay. So there are this is why how the genetic engineering is done. So you develop two different constructs. One is having this the gene for the zinc finger nucleus, which will express this zinc finger nucleus protein and this POC1, and then you insert another construct which will have homology to this region the uh, of the gene where you want to insert your gene of interest. So there will be homologous recombination, and that desired gene can be inserted into the genome of the uh, organism. Okay, there are various steps and quite detailed methodology how this works. So, but the basic concept works on this principle. So the cleavage event induced by the zinc finger nucleus, yeah. So if it finds that there is homology, then this homologous recombination will start to act. But if it doesn't find homology, then this non-homologous engineering pathway will function, and that portion it can have a mutated form of the gene, or there can be other consequences, insertions or deletions. Okay. So each zinc finger nucleus contains the cleavage domain. So the cleavage domain is present where it is present in the FOC1 restriction enzyme, and it is linked to an array of 3 to 6 zinc fingers. Okay. So here they have been shown. It has been shown that there are around 4 zinc fingers. Okay. So this is the FOC1 gene, and this FOC1 gene is attached to 3 to 4 genes of this zinc finger protein. So the zinc finger protein, we had discussed how the zinc finger protein binds to the DNA. So there is histidine residues, which which are present in this helix, which go and bind to DNA region. Okay. To specifically recognize sequences that flank the cleavage site. So this is your cleavage site. This one that is shown in gray. This is our cleavage site. So you design your zinc finger nucleus in a way that it can recognize this green, blue, and red portion. And there are small number of bases, typically 5 or 6, which separate the zinc finger nucleus target. Okay. So you see here that this is designed in a manner, this different zinc finger protein, that this zinc finger can recognize this GGG, this can recognize GGT, this can recognize GAT. So in this way, you can design the construct of this zinc finger nucleus, and you can then transfect it into a mammalian cell. Okay. So this particular zinc finger protein is recognizing this GGG. This is recognizing GGT. So in this way, there are different modifications that enable it to recognize this codon in triplet. Okay. So if the zinc finger nucleus induced cleavage event is resolved via non-homologous engineering, then what will happen then? As I was mentioning, it will result in small deletions or insertions, effectively leading to a gene knockout. Okay, so it will that gene, if you want to remove that particular gene, then there can be different deletions or insertion, and that gene will be removed. Okay, now this approach has been used to establish facile and efficient reverse genetics in Drosophila melanogaster, zebrafish rat, Arabidopsis thaliana, and mammalian somatic cells. Okay. So if the break is resolved by a homology-based process, then what can happen? Then that can enable a modified form of gene to get inserted in your target gene. Okay. In the presence of an investigator-provided donor. So this is investigator-provided donor is the particular gene or the correct gene. If you want to swap a mutated gene with a correct gene, then that correct gene is called the investigator-provided donor. The small changes or entire transgene can be transferred, often without selection into the chromosome. This is referred to as gene correction and gene addition, respectively. Either you can create a gene knockout, or you can create a gene insertion or gene correction or addition, as it is commonly called. So this approach has been used to make novel alleles in Drosophila melanogaster, mammalian cells, and tobacco, and has been used to drive targeted integration in maize, tobacco, and human embryonic stem and induced pluripotent stem cells. So there are different ways by which these are being used. Okay, it requires a lot of expertise and knowledge on how you can delete particular gene, you can insert a segment of gene. Different methods and protocols are there, which has to be followed for such kind of experiments. Okay, now, okay, so this has been taken from that paper only. So this zinc finger protein region. So this zinc finger protein provides a zinc finger nucleus with the ability to bind a discrete base sequence. So that I was explaining in the figure just now. So this zinc finger protein has the zinc finger nucleus. This zinc finger nucleus has the ability to bind a discrete base sequence. Okay. And this recognition region contains a tandem array of cis2 histo fingers. So there are two cysteine residues, and there are two histidine residues, which will go and bind to your. So this is the active site of this enzyme, which recognizes approximately 3 base pairs of the DNA. So in early studies, individual zinc finger nucleases used 3 fingers to identify a 9 base pair target. So each finger will recognize how many base pairs? So each finger will recognize 3 base pair. So if your target gene has 9 base pair, you want to delete or you want to replace a 9 base pair segment or gene with another gene, then what you have to do, you have to create 3 zinc finger nucleases or say 3 zinc finger protein. So each one will recognize 3 base pair. So if there are three, then together they will recognize 3 plus 3 plus 3 is 9 base pair. So total you can, if there are more segments, then more number of zinc finger protein gene for this zinc finger protein has to be created. Okay, so up to 6 per ZFN. So more recent studies have added more fingers, up to 6 per ZFN. Okay, so one zinc finger nucleus will contain around that have been generated. They are having around 6 zinc finger proteins that can recognize even longer segments of the DNA. Now, I had discussed that concept of talents in the extra session. I will discuss it today as well. But just now I was discussing about the zinc finger nucleases and talents. So both the mechanism or principle of their working is based on what? How do they allow for targeted genome editing? The answer is recognizing specific RNA sequences, binding to methylated DNA regions, recognizing specific DNA sequences, and interacting with histone proteins. So they are all nucleases, right? So either they can. So there are two possibilities. If you do not know, then also if you know the terms, the nucleus means they will either degrade what? They will either degrade DNA or they will delete a portion of RNA or DNA. But thus now we discussed that these zinc finger nucleases, talents, they are all specific to, they are designed in a manner that they can recognize specific regions of the DNA and leave them, or they can delete them, or that can be swapped to or replaced with a functional form of the gene. Okay, so the answer is it will recognize specific DNA sequences, isn't it? Okay, so they will recognize specific DNA sequences. So if you want zinc finger nucleases for genetic engineering, then what is required? First, you have to create a construct. Okay, so you have to create a plasmid construct, or it can be a mammalian vector construct, which is having the gene for the zinc finger nucleases. So once you have engineered this construct, then this construct will be transfected into a mammalian cell or any host cell. It will be transfected or into any host cell, and then this host cell will, what it will do, it will start expressing this protein. From the DNA, you will get a RNA, and this RNA will be translated into a protein. Okay, so this is the first process for any genetic engineering, not only for zinc finger nucleases, but any genetic engineering follows this workflow. So you have to design a construct, then this construct has to be transformed into a bacterial cell, or it can be transfected in a mammalian cell, and then this process. So what is this process? The process involves introducing either plasmid DNA or mRNA that encodes a specific zinc finger nuclease into cells or embryos. So this introduction can be accomplished through methods like microinjection or transfection. We will discuss this concept of microinjection in subsequent slide, but transfection we have already discussed. So transfection is what it is. It is transformation in mammalian cells basically. So if you want to introduce a DNA into a mammalian cell, then that process is called transfection. While if you want to introduce a foreign DNA or a vector containing a cloned gene into a bacterial cell, that is called transformation. So once inside the cell, what will happen? The encoded ZFN will be translated, and the resulting zinc finger nucleus pair binds to its designated target in the genome. This binding facilitates the DNA binding dependent dimerization of the FOC1 catalytic domains, leading to a critical step, which is DNA cleavage. And what is the basis of how they work? So these will be required for producing any gene lock-in, gene knockout, and so their ability to induce targeted double-stranded breaks. So both of these nucleases, engineered nucleases, they cause double-stranded breaks and exploit the cellular repair machinery. So these are from the lecture slide. If you go to the lecture slide, then it is explained in quite detail. So how does this? So this is again from this lecture slide. So you see here how this zinc finger nucleases work. So if you want to create targeted mutations in the genome of various organisms, then how you can do that? You can engineer nucleases which consist of zinc finger proteins. So these are zinc finger proteins, individual that are composed of one helix and two beta sheets. These are the zinc finger proteins which recognizes a particular nucleotide base. Okay, or they are recognizing nucleotide bases in triplets, as I was showing in the earlier slide, and a nucleus domain that induces double-stranded breaks at the target site. So at the target site, by this, because it has this fork1 endonuclease, it will form a basically this zinc finger nucleus. It forms a dimer. Only when it forms a dimer, this fork1 will be able to cleave a portion of the DNA. Okay. So to assess zinc finger nucleases in bovine fetal fibroblasts, researchers targeted the 15 base pair signal peptide sequence in exon 2. Okay, so this is the exon 2, which is composed of 51 base pair signal peptide. So by incorporating a splice acceptor sequence in intron 2, they aim to mimic endogenous beta-casein expression. So zinc finger nuclease pairs were designed and screened in a yeast system, and their disruption efficiency was tested in fetal bovine fibroblasts. The ZFN1, ZNF2 pairs exhibited superior cleavage and mutation initiation compared to ZFN3 or ZFN4, particularly at higher ZFN levels. So there are different types of zinc finger nucleases. Okay. So these are basic questions. So the statement that describes the mode of action of zinc finger nucleases and talents in genome editing is what? So as I was explaining, so what are the options? Let's read first. So they inhibit DNA replication, they induce DNA methylation at specific sites, And they introduce single-stranded breaks in DNA. Option D is they generate double-stranded breaks in DNA. So both the zinc finger nucleases and tallins have this fork one restriction in the nucleus domain. So these are only functional, or they can cleave a particular portion of the genome only when they form a dimer. Okay. So this fork one dimer can binds to that cleavage portion of the DNA, and that portion of the DNA is then deleted. So what do? They generate double-stranded breaks in DNA. Is the correct answer. And once this double-stranded breaks are created, then this repair machinery will start functioning in the cell. Okay. So both the zinc finger nucleases and tallins, which are transcription activator. Transcription activator like affected nucleases, they are programmable site-specific nucleases which are used for genome editing. And both ZFNs and tallins create double-stranded breaks at specific DNA location. Okay. Both of these cause double-stranded breaks. Remember this point, and they trigger DNA repair mechanisms that can be exploited for various genetic modifications. So the zinc finger nucleases, what do? They utilize arrays of zinc finger proteins. Just now I was showing that whether if you want to detect or recognize, if you want that zinc finger nuclease to recognize a segment of 9 base pair, segment of DNA which is composed of 9 base pair, then you will require 3 zinc finger protein. So, so there has to be arrays of zinc finger proteins uh, uh, which are utilized and they recognize and bind to specific DNA sequences. So the zinc finger protein binding modules are fused to fork-1 and cleavage domains, which induce a double-stranded break at the target site. So as I was explaining that this fork-1 dimer is required. So two molecules of this zinc finger nucleus will bind that segment of DNA, and then this fork-1 will leave that portion of the gene. And then talents also are transcription activator like effector nucleases, which recognize and bind to DNA. So they also have this fork one restriction endonuclease. They're fused to fork one restriction endonuclease domain. Okay. Both these causes double-stranded breaks. So the initial step involved in employing zinc finger nucleases for genetic engineering is what? So what is the initial step that you have to that has to be done for employing zinc finger nuclease or any type of nuclease? You want that particular nucleus to be expressed in the cell. So what you have to do, you have to transfect the cell with the gene for that particular nucleus. So once you transfect once the cell takes up this plasmid carrying this gene, then the basic molecular machinery will start and it will be translated into zinc finger nucleases. Okay. So what will be the answer? So initial step will not be cleavage DNA at designated target site. So that is a much later stage, a much later step in the action of the zinc finger nuclease, inducing cellular stress response. So the zinc finger nucleus stress response is also a later step when this double-stranded breaks will be created, then this repair machinery will start functioning and it can create a stress response. Initiating DNA binding dependent dimerization. So dimerization is also a later step. So first for all of this, what is required? All of this first requirement is that your gene is getting expressed to produce that particular protein, which is the zinc finger nuclease. So you have to transfer the cells with plasmid DNA or mRNA encoding zinc finger nucleases. Okay. This was explained in the slides as well. So to employ zinc finger nucleases for genetic engineering, what is required? required. The process involves introducing either plasmid DNA or mRNA that encodes a specific zinc finger nuclease into cells or embryo. The introduction can be accomplished through methods like microinjection and transfection. Once inside the cell, the encoded zinc finger nucleus will get translated, and the resulting zinc finger nucleus pair binds to its designated target in the genome. This binding facilitates DNA binding dependent dimerization. So once the zinc finger nucleus goes and binds to its target genome, then what will happen? It will facilitate dimerization. There will be dimerization between the zinc finger nucleus. Okay. Now, apart from microinjection and transfection, what other methods can be used to introduce zinc finger nucleases into cells or embryos? So these are basic questions, isn't it? For any, you have learned about these methods, how you can introduce a foreign gene in any cell. So you have learned about these different methods, electroporation, then microinjection, then transformation, then this that physical by using different physical methods, chemical methods. So centrifugation, by centrifugation, what you can do? Centrifugation is used for what? It is used for sedimentation, or if you want to separate a higher molecular weight substance from a lower molecular weight substance, then you can use this centrifugation. So if there is a suspension of different molecules and you want to separate these on the basis of their molecular weight, then you can use centrifugation. It has nothing to do with introduction of DNA into a cell of your embryo. Electroporation, yes, electroporation is a method. You already have learned about this. And what is dialysis? Dialysis is a method by which you can exchange or basically this will follow the process of osmosis as you have learned. So you want to exchange one solvent with another solvent, you can do this dialysis method. You will follow the process of osmosis and there will be exchange of buffer between uh, or there can be exchange of solvents between two different composition uh, of the solvent. And sedimentation is also nothing has nothing to do with this introduction of DNA into a cell. So the correct answer is electroporation. We have already learned about this electroporation in detail. Let's recap once. So electroporation involves applying a brief electric pulse to the cell. As the name suggests, electroporation is electric field you are applying and which is causing pores in the cell. So it is the application of brief electrical pulses which will create pores in the cell, and this DNA can then enter inside the cell. So you are making competent cells. So making competent cells, there are different methods. One is what? One is the heat kill method, this heat shock method by using calcium chloride. Another is the electroporation method. So this creates temporary pores in the cell membrane and allows the zinc finger nucleases to enter the cells efficiently. The electric field disrupts the phospholipid bilayer, making